Hello everybody, my name is Helge Aasen, and before I start my presentation I will just briefly give you some info about me, because we are an online-only conference. So my main research focus is on ecophysiological remote sensing, field phenotyping and UAVs. I'm with the ETH in Zurich and here with the crop science group. And within the crop science group I lead a small team which is mainly dealing with UAV research for field phenotyping and precision agricultural applications. If you want to learn more about it, then just visit our website PhenoFly.net. One topic of our research is UAV high-throughput field phenotyping of canopy temperature. And this work was mainly done by a master student of mine and me, but also a lot of other people contributed to this from our group, namely Andreas Hund, Jonas Anderegg, Lukas Roth, Achim Walter and Frank Liebisch, and also from the biometrics group at Wageningen University and Research Center, namely Martin Böhr. Thanks for everybody who contributed. Also, the research is recently published in Frontiers in Plant Science, so if you like, you can read more about it in this paper. So why is canopy temperature important? Well, it's related to water use and yield formation in crops. And it would be very handy to have a high-throughput field phenotyping method to screen genotypes for these parameters. However, canopy temperature is also influenced by fluctuating ambient conditions, namely temperature, wind, but also illumination conditions. So we need to take care of this if we want to have a reliable sensing procedure. Besides, as in every field phenotyping experiment, we also have in-field heterogeneity, which might mask the genotypic effect, which we are ultimately aiming at. And then also data processing workflows, so the way we handle our data and process our data, may also influence the apparent canopy temperature. So the goal of the study was to develop and evaluate a UAV-based high-throughput field phenotyping workflow for canopy temperature. Our study was carried out at our plant research station in Eschikon, where we have the setup here. In total, we have we had around 360 genotypes of wheat in 2 repetitions in these 2 plots, which you can see up here and down here. The crops are managed in a typical manner for Switzerland, and no treatments are applied. The whole station is equipped with ground control points, so we can easily carry out multi-temporal studies. And in addition, we also have a local weather station here, which records the typical ambient information. Our data was collected by our self-developed multi-sensor payload on a DJI Matrix, which was flown on a DJI Matrix 600. And for this study, the most important component is the thermal camera, which is a FLIR A65, which we mounted into a housing which is temperature controlled. Besides, we also fly 2 spectral cameras and a 12 megapixel RGB camera on this payload. So the first thing was to develop a flight protocol and a data processing workflow. For the flight protocol, we flew with a very high overlap at 80 meters. The overlap was about 90%, so we could compose a lot of images to one auto-mosaic in the end. And the camera records raw thermal images, but the camera is radiometrically calibrated, so we could transfer these into temperature readings in Celsius. After this conversion of the images in temperature degrees Celsius, we imported the image into Argisoft Photoscan and ran them through a typical photogrammetric image processing workflow based on structure from motion. One important component there was that we used specially designed thermal GCPs, which were actually visible in the thermal automosaic to do the georeferencing. In the paper, you can learn a bit more about which blending modes we used, but in the end, the best blending mode for this purpose we figured was the average blending mode. With this average blending mode, we then generated georeferenced automosaics. And here you can see one example of this automosaic. So the color scheme shows the temperatures between 28 and 40 degrees. And as I said before, we were flying at 80 meters, and this gave us a ground sampling distance of about 6 centimeters. The next step was then the feature extraction. And what we found in our automosaic is that we had quite some strong border effects where the paths between the plots and the plot kind of interfered. So what we decided in the end, the plots were 1.5 meters wide, but we just took the center part of the plots, which was about 50 centimeters of the plot center and about 1 meter long. From these center areas, we extracted then the canopy temperature for each plot. In the end, we are interested in the genotypic data and the genotypic effects. But as mentioned before, often in field phenotyping experiments, we have influence of in-field heterogeneity. And this was also the case for us. So the automosaic which you see here is generated in the end of June 2018, which was quite a dry year for our in our region. So besides of the genotypic effect, we also, which can be seen here, that the plots have different temperatures next to each other. We can also see some broader patterns in our field. So for instance, up here, we have a kind of a high temperature island in the middle. Also down here, we can see some spatial trends in our field. This is coming from the soil which we have below our field. But also, in addition, the management can also influence field phenotyping experiment, which can be seen here, that there might be a management effect along the rows. And we need to take care of this. Moreover, also the ambient conditions can influence our measurements. So down here you can see an except of a flight where we had not 100% stable conditions. They were visibly almost stable, but still, if we look close here, then we can see that there are some steep transitions from within the field. So there's one here, and one here, which is quite apparent. And this then comes from the interaction between the slightly changing ambient conditions and the flight pattern. Because if we look at the flight pattern which we used, then we flew in these directions, of course much denser, but this is just to show you how we flew. And well, this then matches up with these stripes. So these images are basically taken from one flight line, and this is taken from another flight line. And these are also effects which you need to take into account. And last but not least, also the bidirectional reflectance distribution function, which is also apparent in temperature data, also needs to be taken into account. But I will not touch this topic here, but if you're interested, have a look at the paper. So how then can we correct for all these effects? Well, it's a couple of things. First of all, due to the data processing modes, and this is discussed in the paper. But also, we applied a special correction procedure to correct for these effects and to get the genotypic effect in the end. And the procedure we used is the SPATS package, which is correcting for spatial heterogeneity in plant breeding experiments with pea splines. And you can find the citation down here in the slide. After applying this spatial correction algorithm, we then got the corrected canopy temperature for each plot, but also the genotypic information. So here you can see the results of the SPATS package, and on the right you can see the results for the in-field heterogeneity which the SPATS package gave us. And you can see that for the spatial trend, SPATS package very well estimated the spatial trends which we have in the field, and then also could correct for this. So to sum up, basically our plot temperature is coming from the genotypic effect, but then we also have spatial trends coming from the soil background, for instance, and also from the changing ambient temperatures, which also go into the spatial trend. And then, of course, in the end, if you have the optimization process, then there are also some residuals. But we are interested basically in the genotypic effects. And if you have applied this procedure, then we can also look at the repeatability of our measurements of our procedure, or the heritability if we talk about genotypes. And this was then the next question. So when is the perfect timing on the day to fly? And here you can see that we carried out flights throughout the day, starting from 9:30 in the morning, 10, 11, 12, 30, 2 o'clock, 3 o'clock, and 4 o'clock. And down here you can see it was a very, very nice stable day. The temperature increased throughout the day. Each stripe here is referring to one measurement flight. And also the photosynthetic active radiation was very nicely distributed. If we now look at the heritabilities which we gained from these different flights, then we can see that afternoon they were highest. And this is also kind of what we see already in the pictures, that in the early afternoon, the differentiation between the plots is also visually the most apparent. Another thing which we can see is that if you compare the values, the heritability values without any special correction, these are these stars down here, then we have a very low heritability, and the heritability increases significantly if we go up, if we use the special correction algorithm. So the last aspect of our study was to compare this developed UAV automosaic approach to other approaches. What we found is that the UAV approach is actually quite feasible for field phenotyping experiments, because the setup costs are quite low, and especially the running costs per field area are very low, because the hardware of the UAV is cheap, and also to cover the whole field it takes about 10 minutes, and then we have screened about 750 plots. Well, of course, you need to set up the UAV first, which takes some time and also some knowledge. But after this, the effort of the measurements are quite low, and the plots sampled per time, it's also quite fast. A big benefit is the flexibility of the sensor setup, because basically you can go wherever you want with your sensing system as long as it runs, or as soon as it runs, then sample wherever you want. However, of course, if you want to have georeferenced images, you also need to prepare this by putting ground control points to your field, which is an additional effort, but just need to be done once in the beginning. And what's also quite nice compared in particular to ground-based systems is that this is also applicable for tall crops, because the UAV can basically fly very high, and there's no limitation there. With this, I would like to sum up. So what we found is that a spatial correction workflow is needed to really get genotypic estimates, not only because of the typical in-field heterogeneity, but also if we have slight changes in the atmosphere, we also need to correct for this. Then optimal timing for the genotypic differentiation by means of heritability was in the afternoon. And compared to other approaches, the UV-based high-throughput phenotyping is very suitable to screen even large populations of genotypes in a quite fast manner. Some things which I didn't talk about too much now in the presentation are the angular effects, which influence the apparent canopy temperature, and also the influence of the different processing modes. So if you want to learn more about these things, and also want to read a bit more in detail about the research which I just presented here, then please refer to the paper. You can find the citation down here. So thanks a lot for your interest and for your time. And if you like, and if you want to learn more, look at the paper or just get in touch with me. Thanks a lot, and hopefully see you soon again when it's possible. Bye.